Thank you.
So we, as you know, we held a brainstorming way back in January time, and we're now starting to work on some of the ideas following up from that. We'd also like to start thinking about some structures that we might want to do around these literature compounds that we've now made, and we've got some information from the screens. I think we plan to have some more brainstorming sessions, and that would be particularly useful if we get some crystallization results from those compounds. As I think Jeff mentioned, we were also running some stick profiling, which we hope to get the results as soon as possible. So just to go through, just talking about these literature compounds. So on the left-hand side, we've got the Merck compound, from the Merck patent. I understand that the RBC results are available, and I haven't managed to see them yet. But looking at the data from the patent, they should be pretty potent. I guess the data from the nanobret assay, which Alex has just shown, is on there with structure. That first compound was only moderately active. But on the right-hand side, we've got the blueprint, compound blueprint patent. And in particular, we've got this one compound 3007, which looks pretty potent in the cell assay, and I'm imagining it's pretty potent in the enzyme assay. And it looks like it's got good selectivity as well. As I mentioned already, it would be really nice if we could get some crystals of those compounds. So it's interesting that this compound has got that epoxy on the pyridine ring, and I think we've got another one of the compounds. I can't remember whether that one's got the epoxy, or there's certainly a compound that we're making that has not got the epoxy on there. Yeah, so that would be interesting to see whether that's actually really key. Yeah. Yeah. And these compounds look like they sit quite differently when we did some overlays. It was difficult to see exactly where that type of profile ring would fit, so that's why it's really nice that the crystal structure of it will really help us in understanding what's happening in that region of the molecule, or of the protein. We also had a look at a couple of compounds from the Novartis patent, and the one on the left, 3003, shows really good results in the nanobret assay. The compound lacking, the related compound but lacking the hydroxy group, is a bit less active, but it's not clear at the moment which is the feature there that's actually adding to the result. So we have also made the compounds with the methyl group where the amino is in those, because at the time that we started those syntheses, that was of interest. But it may be worth thinking about some other analogues, or using some overlays, again, using some overlays from these structures to our own our own pyridine series, see what we might be able to use from that structurally to design some new compounds.
Yeah, even the 4-pyridol.
Yeah.
The 4-methylpyridol.
Yeah, exactly. Yeah. Sure.
Yeah.
Yeah. Yeah. So in terms of some new targets, so these are things coming from our original brainstorming, and in the original brainstorming we probably had a methyl in the 6th edition, but we've taken that away. As I think we've all said along the line, is ideally we'd love to get rid of that trimethoxyphenol. So from some of the early work, the epoxyindazoles look to be somewhat interesting, perhaps not quite as good as we might like, based around how those appear to sit in the proteins. Then we were looking at replacing the compound, replacing the indazole by benzimidazoles, and the first compound on the top left marked as A has been completed, and that will go with the next shipment. That's just a straight benzimidazole, which is effectively replacing two of the oxygroups. So it'll be interesting to see whether that's successful. Looking at some other types of molecules, you can imagine replacing the phenol by heterocycles, such as pyrosols, and we've completed the synthesis of the hydroxyethyl pyrosol there, which from our modelling shows at the bottom there appears to pick up an interaction with glue 248. So that's compounds actually in purification. Hopefully we'll be able to get that into the next shipment, but that depends on timing, etc. So we're making some progress with these targets.
So we normally have a water-mediated hydrogen bomb there.
Yeah.
I don't know whether your computational advisor can tell us if we should keep the water or displace.
That's what Sylvia's actually been looking at, to see exactly whether that's a happy water or a whether we need to actually interact with it, or if you displace it, you need to have a different in that you need to have quite a strong interaction there. So yeah, I agree. It is about. So a lot of the docking that we've been doing, we do first of all in dry, and then we map the waters on using a water map program, and then we see whether those waters are actually displaceable or whether they're essential. So we've also just started to look at some of the core replacements. So based on some of the structures like the ICR, phenazolone type structures, thought it could be interesting to use a pyrimidinone, such as those compounds on the left-hand side in the yellow box. And we've just started the synthesis of that compound marked C. And this also sort of fits in with that idea of having a group in, like the methyl group, in that 4 position of the pyridine. So that compound synthesis has just been started. On the right-hand side in the blue box, we're looking at alternative core replacements, synthesis of target D, where I think you mentioned earlier, Jeff, that having the pyridine where the nitrogen is where the methyl is, D-methyl. So putting the nitrogen next to the pyridine nitrogen in the we're getting pyridines and methyrazines mixed up, but the compound shows that marked as D there. Will be interesting to see how that functions. So I've had a few more new ideas. So it would be nice if we could replace the transoxyphenol with something perhaps non-aromatic and see what having a bit more 3D character in there that could improve the discount properties. While I accept that having a primary amide in there is probably not ideal from a discount properties perspective, if we can see whether getting into that sort of region, we can then replace that amide by some other functionality. So compounds like this pyridine amide could be interesting. Still only has 3 H1 donors, so if you can remove the one on the right-hand side, or possibly put another group on the pyridine to get an internal hydrogen bond so that you tie one of them up.
No, absolutely. I think, if we are able to get X-ray structures and use that information to guide further SAR, right?
Exactly.
Yeah.
So the other thing that was quite interesting was sort of thinking about if substitution in that 4 position is tolerated, can we then actually use that rather than having the group coming off the 3 position of the pyridine come off the 4 position? And we did some docking, and this amide compound, again, if that's using amide, some nice interactions. It picked up some really good interactions up in that water-mediated region, and several water-mediated interactions with different groups there. So this could lead us into something structurally slightly different.
Yeah.
And if you can make something like this and it's active and we can get crystal structure, that's really going to help us. So the final slide is just something that Sylvia has been looking at, is considering what else can we do to look at a lasting interaction. So replacing that trimethylphenol, and this is being done on a sort of fragment hopping, generating a huge library. trying to build that library and dock it and see whether, and look at SysChem properties and see whether there's any completely different functionalities that we could use rather than just having to come up with the ideas and dock them and come up with another idea. This is a more a CAD-based approach. So we started looking at this. It will take a bit of time, obviously, to refine, but it's something that we're looking at.
Well, it's great. There's some hits on the right-hand side of the slide, or just
that's just some illustrations of how they go. No, we haven't got any hits yet. It's just she's only just started in the last few days. So, yeah, I think that's my last slide.
Okay, fantastic. Thanks. So I think it's David Smeel. I think David Smeel. David, you want me to drive it?
Okay, can you hear us?
Yeah.
Okay, go for it, David.
Great, thank you.
Or do you want me to transfer control to you?
Or if I can just say next slide, you guys can handle it. Yeah, okay, perfect. Okay, so these first four compounds are kind of the foundational compounds for everything we've been doing after. And I've taken all of the data that we have to date from RBC and from the cell-based assays, and I've aggregated them all underneath each compound, so there's a lot of information there. But just to point out a couple of things to start, using 1062 as an example, the small number 2 in brackets is the second batch of that compound that we made, and that just illustrates that we're seeing consistency in the screening at RBC between batches, so that's a good sign. You'll also notice that there's an ALK4 IC50 number now, which we've been getting in the last couple of rounds from RBC. And the nice thing about it is that ALK4 does seem to be paralleling activity on ALK5. That's to say that if we're not hitting ALK5, we're also not going to be hitting ALK4. So that's good, especially based on what BI was saying, that there's a high risk profile on ALK4, but it seems that we'll be able to avoid it for the most part. For the nanobreath, the values are there, and there's also the values for the luciferase. For the instances where we have the luciferase both on the ALK2 and ALK5, I've calculated the ALK5 to ALK2 ratio for the selectivity index. I have not done that between the two types of assays because they don't necessarily correlate. What we're finding is that for the luciferase, the compound's actually a little bit more active than in the nanobreath. So you can just kind of gauge just visually in terms of what the selectivity is, but I haven't added the index specifically. 1071 was a compound that we've been looking at for a while, whether we could actually eliminate that middle methoxy of the trimethoxy group. And it seems that potency is retained on the biochemical screening, but unfortunately this compound and a couple other follow-ups I'll show in the next slide, they don't have spectacular cellular activity. So I think that's the kind of the dream of getting rid of one of the methoxys is not going to come to pass. I think if we're going to be stuck with the trimethoxy group, we're going to need all three methoxys rather. 1055 is another compound that we've kind of dropped over time because we've realized that the potency in the biochemical and in the cell-based is not great, nor is the cell activity. So we're kind of moved away from the series with the methyl group at the 2 position. And instead, we're focusing on the 2009 series, where we have the methyl group in the 4 position, which, as you can see now on multiple retests, is showing a really good window of over 100, in many cases over 200-fold ALK5 over ALK2. The cell-based assay is kind of in that 40 to 70 nanomolar region, so that's quite good. And so we've been kind of focusing on that group as the pivotal group that we add to most of the compounds going forward. Just a note now, we're about 100 compounds into the series, and on the next few slides, I'll show you just some of the more key compounds. Much of what we've done around the head modification, as you see on this slide, has not worked, and that's involved eliminating one of the methoxy groups, changing positions, varying substitution patterns, etc. And all of that has been largely detrimental, with the exception of these compounds shown here. So here's the 1071 parent again, and when you add the methyl group to it to make 2059, you see the biochemical potency is quite good. The cell activity is substantial, but again, the cell-based assay not quite where we want it to be. So again, I think this is a series that, or at least this dimethoxy motif is something we might abandon. The last compound, 2053, is a little bit interesting in the sense that if you add back a fluorine between the two methoxys, you do have a very potent compound biochemically, and you do start to claw back some of the cell-based activity. But again, it's a little bit suboptimal for us. So again, probably park this idea for now. Next slide.
It's interesting that fluoro brings back potency against ALK4 and ALK5.
Right, yeah, which is hard to
I
rationalize.
guess this one also has that T4 methyl, so.
That's right. Yeah.
Yeah. Okay.
So here's some different modifications. This is totally trying to drive away from the trimethoxyaryl, and this is the amide series. So 2017 was the early hit based on this idea or this chemotype. Pretty decent activity on the biochemical assay, but unfortunately we didn't get a cell-based reading. We didn't make enough compound. But a variant, 2096, was made, and you can see it's very potent on the biochemical and it's also showing quite good activity in the cell-based setting. And it's actually comparable to 1062, which is the parent, and 2009 also. So this may be something that we may want to look into further and evolve as a parallel series and a different type of head group. And you can imagine taking 2096 and of course putting the 4-position methyl on there to see what happens. One compound that's still yet to be tested is 2114, and we'll see what comes of that. So again, this series of potential, we can branch off and do some more exploration around it if you want to. Next slide, please. So one of the things which has been kind of consistent for us and which has proved to be quite decent, not only from a kind of activity perspective, but also from in a biochemical activity perspective, but from a cell-based perspective, is variation of the southern group to a variety of kind of tethered pyridines, a variety of pyridines, either alkylated or unalkylated, and also a pyrazole core, as in the case of 2044. And all of these compounds, broadly speaking, in the cell-based assay on ALK2 range between 30 and 70-odd, 80-odd nanomolar, which is not bad. 2044 was the best in the series, starting to push the kind of 30 nanomolar range. And they've been consistent in terms of, you can see small structural changes have essentially preserved that activity across the board. And it's these compounds which we've been taking into the PK and into some of the work that Jerome will show in a few minutes regarding the activity on the DIPG lines. So these have kind of been the workhorse compounds that at least establish some of the PK and CNS penetration and some of the DIPG line work that we're trying to do right now. Next slide, please. So working still in that kind of southern solvent front exposed section, we've made a variety of changes there. Again, nothing's been spectacular in terms of helping activity and selectivity, except for these 3 examples here. 2085, perhaps unsurprisingly, is again active in that 50-odd nanomolar range of the cell, given that we've made a very small chlorination in that ortho position. So kind of to be expected, I suppose. And what's interesting is that 2087 and 2089 are really starting to show great biochemical potency, and they're also starting to be quite potent in the sub-15 odd nanomolar region on ALK2 in the cell-based assay. And so that's pretty encouraging. Maybe we're getting a little bit more cell permeability or whatnot, and this makes these compounds about 2 to 5 times better than the parent and 2009 in the cell-based setting. And one of the motivations of making these smaller compounds was to see if we could get some better CNS penetration, perhaps, going forward, if these compounds do in fact show good PK and other properties. You'll notice that both 2087 and 2089 don't have the inside methyl group, and so those are in plan and should be made shortly. And also 2113 has been made, and we're awaiting testing on that variant to see if we can make that an aryl system entirely. So again, work continues around the series with these smaller southern portions. Next slide, please. Another idea that's kind of we've been looking at more recently, and Dave McLeod has begun to work on this, is some work inspired by this JMed paper from a couple of years ago, where they put these ALK inhibitors, this is ALK1, into the clinic. They're very potent, as you can see, the picomolar compounds, and they show really good selectivity against a pretty diverse panel of other kinases. And so the idea was to adapt some of these motifs and ideas into our series. I mean, the central arrow there shows you that we can either preserve that structure in its entirety, or as the top arrow shows, we can take that aniline motif and see if we can piece it together with our central pyridine ring to create structures of that type, thereby having a kind of bidentate hinge binder possibly. And also you can think about the compounds at the bottom where we keep that pyridyl directly attached to an arrow group, but now we have that analytic linkage in a different position to see if that's tolerated. So these are some of the ideas that we're currently exploring, and hopefully some of these compounds will be submitted in the next screening round at the end of June. Next slide, please. So this is a structure you've seen many times now, the parent 1062 with all its attendant data. And the idea was that we would like one of these compounds with the internal 4-position methyl crystallized. And if you go to the next slide, please. This is the compound that Alex was showing. This is M4K 2044. So this is the first compound with the methyl group on the pyridyl, which we've been successful in crystallizing. And again, as Alex showed, not too many surprises in terms of how the compound is arrayed in the pocket. And it's going to be interesting, though, to see once if ALK5 indeed can be crystallized, to do the direct overlays of ALK2 and ALK5 using a compound like 2009 to see if that methyl group really does make a difference between those two particular targets. Next slide, please. And that's it. I thought there was more.
Okay. Thanks, David.
Yeah.
Yes. Okay, Jerome is in the room, I believe?
Yep.
Yeah, okay.
Can you hear me well?
Yeah, yeah.
Okay, good.
Yeah.
Okay, thank you. So we started testing some of the M4K compounds on DIPG cell lines, and here I'm just showing you an overview of the 3 cell lines that we've been using. So 2 of them carry the ACVR1 mutation. Both of them is glycine 328, which is mutated, and then we have another cell line, DIPG6, which does not have an ACVR1 mutation. So those are cell lines that were obtained from Michel Manget's group at Stanford. One important thing to note is that these cell lines are very different in the way that they grow, in the way that they look like, and they have very different proliferation rates, as you can see on this table here. So we, try to play around a little bit with the number of cells that we place per well in the assay to get good data, but just keep that in mind when interpreting the results. So the experimental design is very simple. We placed the cells on day 1. The next day we had drugs, 10 concentration assay. And then 4 days later we assayed the cells. This is the ATP-like, it's like cell titer glow. So if you go to the next slide, you can see the results of the 6 compounds that we've tested so far. So, I mean, you guys can see probably some maybe interesting things in there. It seems like in general, the DIPG21, so the red line, responds better than the DIPG4. You also see the DIPG4 kind of plummeting at lower values at the higher concentration. But keep in mind also that these cells proliferate much faster. So, if you envision a scenario where you have like the 100% cytostatic effect, then you would get down to a much lower value with the DIPG4 than with the DIPG21. And the DIPG6 is often not very much responding to the compound. So yeah, so that's what we, yeah.
Which one's which? Which was the blue is wild type ICBR1, green and red was the
Yeah.
so 6 is the negative control. You should see no. Okay, thanks.
Yeah. So yeah, so this is, so again, yeah, so you have to take into account that because the 4 cell lines grow much faster. You know, even if you just have a cytostatic effect, you would get a more, you would get a lower value on the assay readout than DIPG-21. It seems to me that the DIPG-21, if you look, for example, at the 62 compound or the 44, where you see kind of it reach a plateau around 0.5, that's probably like cytostatic effect, although we haven't looked at that directly. Whereas like the other compounds that you see it getting lower, maybe there's some cytotoxicity as well. Yeah.
I guess from my perspective, I think, you know, I think it was interesting, this data. I think, you know, it's nice to see that we're seeing some effects. 2058, the amino series. So I thought that that one doesn't make what we see with the other, right?
Yeah.
Right? It's about the same level of potency, right? That's 30 nanomolar in the nanobreath. Not seeing the effect on the side, so
It's less selective of outside,
it's much less.
more dirty,
More dirty.
distinction.
There is, yeah, but I mean, I would expect it still to hit a target and have an effect on the population, right? It's not a solid. Maybe it's worth retesting that couple. That's a good question.
So could that partial inhibition actually, could that be the ACV dependent? I mean, is it a homogeneous population, or that could be a dependent APG6 pool, and then you actually have a resistance pool that left? Maybe that's the actual ACV phenotype that you actually have for that particular cell type.
Yeah.
These are not the cells that are left.
Yeah, it's possible. Yeah, there could be some heterogeneity in these cells. That's for sure. I mean, these cells are. I think all 3 of these cell lines were taken from autopsy at autopsy, so they're very advanced tumors. These are from patients that have been treated with different things, at least radiation, but maybe other things. And so there could be some heterogeneity in the cell population as well. Yeah.
So you could see, I mean, if you look at the compound 2045, which is actually affecting the negative line, you could see now it's actually starting to be more toxic.
Yeah.
Those are the 2 cell lines.
So if you do, this is 6 days after, right?
Yeah, 4 days after the drug.
Yeah. So if you do go longer, do you think you'll have more drops in the cytostatic?
If that's possible, I think that if you let the cells, you know, if you let the cells grow more and then you keep the drug on, I would think you're probably going to have a similar profile, but that you're basically aligned with it, at least for 21 and probably 6 as well would go down. Yeah.
I think one thing as well, Jerome, is, you know, I mean, we've given you, a bunch of compounds that we're spreading a potency, diverse potency range in the nanobreath. And, you know, I think if we start to see some of that SDR in this as well, that, you know, we start to have more confidence in the results. I think all of these compounds are pretty similar.
What might be, what's the different efficacy between inhibiting the BMP SMAD pathway and inhibiting the right important downstream effects? So compound the bottom right. You know, there's something N equals 1, you know, 1 experiment, let's see. There may be something in what?
Yeah. Early days. I think the results are encouraging.
Yeah.
The chemical effect, you know, knock down one and you stimulate the other, and vice versa.
Yeah.
I think one of the next steps for us will be to maybe take, you know, a couple or 3 of the compounds that look the most interesting, also maybe with the PK and stuff, and then look at target engagement in those cells, you know, see if we can affect phospho-SMAD, downstream gene expression and things like that.
Yeah.
So if you go to the next slide, I think I have a table. Yeah, this is a table giving you the IC50s. I mean, again, like in many cases, it seems like the IPG-21 responds substantially more than the IPG-4. But, you know, keeping in mind this heterogeneity and that these cells are are derived from different patients and from different tumor stages and treatment backgrounds.
Seemed like with 2009, it was 21 was the more sensitive one. Maybe I saw the graph. It looked like it was the other way around, but.
Yeah, I think the 209 is the one that maybe show better for 4 than 21.
Oh, okay.
So, yeah, this one is a bit intriguing in that way. Yeah.
Maybe we should also include the lead compound that we started to use to drive the program that we put in vivo to see that reach effect. It would be nice to benchmark a lot of these.
Right. Yeah.
That should be.
You're talking about the ones, the methyl group in it.
Yeah, the sharding compound.
Yeah. Yeah, that's a good idea. Yeah.
Yeah.
Did you guys say in broad terms, this is, I've maybe seen before, rather encouraging, that you have an ALK2 inhibitor and it kills selectively? I mean, these cells might have just said, I don't care if you put an ALK2 inhibitor on me. I thought a whole whack of other ways to get around it. And so, and the cell lines with the mutation are responding to different degrees, albeit, and the cell line without a mutation not responding. And that's pretty clear.
Yeah, well, I think it's really good. Yeah, encouraging. Yeah, I'd like to see more as it follow the SAR a little bit better, but in a more early stage, N equals 1. Yeah.
This is Javan Nazarian, DC Children. I have a question. So we couldn't actually hear most of the conversation that is going on in the room. I guess there's too much background noise. But the question that I had was, have you tested this drug in combination with HDAC inhibitors, or would you be interested in working with us for your lead compound to be tested on DIPG cells that are either ACVR mutant or not, or even histone 3 wild type in combination with HDAC inhibitors in vitro and or in vivo?
Yeah, I think that's a very interesting idea. I mean, of course, this is early days. We just did those assays over the past month, so we haven't combined them with other drugs. But given that panobinostat right now is in clinical trial, we're eagerly awaiting results of that. At least for the early stages. So it could be extremely relevant to combine that, combine these one or a few of these inhibitors with panobinostat and see if there's, synergy or things like that.
Yeah, I think it's an interesting idea. I think we need to be sure that what we're combining with the HDAC inhibitors is truly on target, and that the results we're seeing are really related only to ALK2. I think we need to generate more confidence in that before we embark on combination studies.
Well,
but certainly down the line, something
we're not restricting anyone can have a compound to experiment, right?
Sure, yeah.
The caveat emptor, it's not characterized well enough. And the reason people are going to clinically is because they can, not because it's a very good idea. And so potentially piling on pain to any recipient of that moment, because it's not a science-based experiment to put an HDAC inhibitor into a child. It's a Hail Mary because there's nothing else we can do. An EZH2 inhibitor would potentially more down. But again, I don't think it's going to stop anybody from doing anything with these.
No, we're like to anyone who's interested in
just that the reason they went into the clinic with HDAC inhibitors because they could, patients were dying and it was approved, not because they threw a kill of something in vitro and it's effect. It doesn't seem proper to me, any opinion.
Okay, so if you move forward one slide and then the next one, which is my last slide, I just wanted to update you briefly on the attempt to generate xenograft models. We know that several of these cell lines, at least, are supposed to be able to engraft well in immunocompromised mice. So I've tried to do that here. And I have two cohorts of mice going on. This is the results from the first cohort. So far, only the DIPG4, which is also the fastest growing line in vitro, has been given tumors. It takes, you don't see the axis here on the presentation, but it takes 2 to 3 months. So maybe there's a bit of optimization required there. So I recently repeated as well with the DIPG21 and doubling the amount of injected cells. And hopefully we can get some reliable models, that doesn't take too long, I guess, to test some of the compounds in vivo once we have an idea about the brain penetration. And that's it for me.
Okay. Any comments on that, Martin?
What's slower death than the other lines?
Yeah. Like 007?
Mm-hmm.
Yeah. Yeah. Interesting, though. Yeah. Okay.
Did someone mention earlier that there is a plan to test the blood-brain barrier penetration of these compounds?
Yeah, we have compounds that have gone into those experiments, and actually we just got results today. I think we have some encouraging results. I'm not going to show them until the next meeting, I think, because we want to evaluate them a little bit more. And I think, it's only exposure data at this point. We don't have any information about free fraction in brain versus plasma. I think, to truly prioritize compounds, I think that will be important. But certainly we have data that looks encouraging. We're reaching high micromolar exposure in the brain with at least a couple of the compounds. And it looks like the KCO2 data that we're generating correlates with what we're seeing in the brain in terms of compounds that show efflux in our KCO2 assay at OICR tend to have lower exposure, although it's a much lower exposure in the brain, almost zero. But it's only one compound, and so, I think we need to get more data before we generate too much confidence in that. But so these compounds, at least some of them, are getting into the brain. Yeah.
And this is by analyzing the brain postmortem.
Yes.
I think, right?
Yeah.
So we have in vivo models where we have microdialysis probes sitting in the brain stem, and we'll be happy to work with you and test your compounds. We will be sending you the washout from the probes. Then you can test to see how much of the compound is in the brain stem in the drug.
Wow. Okay. That sounds great. Yeah. 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 Okay. I mean, please, yeah. Let's talk about that maybe a little bit more. Maybe.
Sure.
Offline or why don't we have a conversation early next week when we're back in Toronto, if that's okay, and just talk about
how to transfer the compound and make sure that everything's in line.
Makes sense. Sure.
Okay.
Thank you.
Thanks. So I think it's me now. All right. So we're moving through this a little bit faster than anticipated, but I think that's okay. I know you guys are just starting your day over there, and you've been able to miss the annual fire drill, which, I think we'll try to schedule a meeting the same day next year. But here it's getting closer to the end of the day, so I think finishing early will definitely be okay. So I think that, we're generating a lot of very interesting compounds. I think, as David Smil said, I think, we're up over 100 compounds now into the SAR. I think things are going very well. I think that, we're starting to gather more information about what's driving the hinge binding, how we can alter the solvent side binding group, or the southern portion, as David calls it. And certainly the trimethylphenol has been challenging to replace and to maintain suitable cell-based potency, at least. I think we've had some success in maintaining intrinsic potency, binding potency, but keeping the cell-based potency has been an issue. But there are compounds out there that don't have that. So, you know, I think that we just need to keep trying. But the PK has generally been pretty good for these compounds, and I'll show you some of that data, even with the trimethoxy. Now, that's still, you know, we may run into a flag later on down the road with maybe potential reactive metabolites, with, you know, we're not out of the woods, but, you know, I think it's certainly, they're not being rapidly metabolized, and preventing absorption.
Can I ask a question, please?
Yeah.
So I remember writing the grant, and I remember Tim Wilson looking at that thing and going, oh my God, stay away from that. But then we keep putting it on, and now we're getting good brain penetration, and it's killing the cells. They're a single. So what is the actual problem?
Again, I think it's a concept. Generally, the trimethoxys, the more methoxy groups you add on an aromatic ring, it becomes more electron rich. You can you can demethylate those very easily. So a lot of times they get rapidly metabolized. And then
treated,
result,
sulfated, or they get.
It can result in a phenol, which can be glucuronidated, right?
Okay.
So it can which can be a reactive metabolite. So, you know, I think there are issues, potential issues. You know, the metabolism data we are generating is only phase 1. You know, only oxidative, but they are getting rapidly absorbed, and you know, we have good exposure. So even phase 2, at least in mice, doesn't seem to be an issue for these compounds. So that's why we didn't completely eliminate it, right? We have a serious effort to replace it, but you know, we are continuing to make molecules with very subtle modifications around that original scaffold. So the chemistry strategy that we've adopted to try to push these things so far has been, like I was saying, a real focused SAR around the lead molecule with subtle changes to try to improve the potency of the 214-117, but maintain the selectivity profile and the PK and blood-brain barrier properties that was observed in that original compound. We predominantly moved away from the 2-methyl group because we haven't been able to generate potent compounds that incorporate that, and we've moved more into doing SAR, or at least initial SAR, where we have the 2-hydrogen, or now the 4-methyl as well. And again, like we were talking about, we've been trying to replace that trimethoxyphenyl with the potential metabolic liability. It's been challenging, and you know, it has been well tolerated in vivo, at least in several circumstances so far. And you know, it seems like that region could be the key to some of the kinase selectivity as well. Certainly for ALK5 and potentially ALK4. I think there's some water-mediated hydrogen bonds over in that area that, you know, if we can pick those up or break them in ALK5, then, you know, we can alter the selectivity profile. So we still continue to work in that area, but it has been challenging so far. I think, you know, Sue showed some very good ideas and interesting ideas around, you know, altering that hinge binding motif. You know, just a pyridine probably doesn't form the strongest interaction with the hinge, but, you know, adding a methyl increases electron density, makes us more basic. Is that causing an improvement, right? I mean, I think that there's some other ideas around that I think David and David on our end have in terms of modifying that core to try to increase electron density and improve that interaction with the core. So we're continuing to follow up on that. And then, you know, certainly this area, the solvent side binding group, has been, you know, relatively easily modified and still maintain reasonable potency. But, you know, certainly we can play with the properties, the physicochemical and the admin properties of the molecule by tuning that area of the molecule. So I just wanted to kind of reiterate the molecules, or re-highlight the molecules that we had selected at the last meeting to move into mouse PK experiments. So 1055 is, again, the lead compound, the 214-117. 1062 is where we remove the methyl and we have a hydrogen. 2009, we move the methyl from the 2 position to the 4 position, get about a 5-fold improvement in potency, and still have pretty good selectivity against ALK5, and it's metabolically stable in the Keiko2 profile you can see here. It's essentially 1-to-1. And then a couple of modifications of 2009 on the solvent side, the solvent binding side, where we remove this nitrogen and make it a carbon. This compound is also essentially equal potent, seems to give a little bit better selectivity, and again, is stable, although this compound does show a little bit of efflux. So I think that's interesting, and I'll talk again about this. And then M4K 2045 that incorporates substitution on this nitrogen to an isopropyl, where we lose a little bit of potency, but again, overall the profile is quite good. So we moved 1062, 2009, 2043, and 2045 into in vivo experiments. There was one other, 2044, which was the pyrazole-containing compound. I'm not re-highlighting that because it did not show good exposure, so I'm just essentially ignoring that result. We won't go there again. Plus, the slide is pretty busy as it is. So, so these are the mouse PK results for these compounds, and I'm comparing against M4K 1055 or the 117. Now, keep in mind that the data for this compound was generated previously in a different experiment, but at 5 mg per kg. So I've taken the exposures generated in that experiment and just simply doubled them. That may or may not be accurate, but just as a comparator, I've done that. But the bioavailability numbers still should be comparable. So 1062 with the 2 hydrogen showed relatively rapid clearance compared to the 2-methyl compound, and I think that correlates fairly well with the mouse liver microsomes. I think overall the exposure wasn't terrible, but it was certainly much lower than with the 2-methyl. Moving to 2009, though, the 5-fold more potent compound, it's essentially equal, has good stability in mouse liver microsomes, good TACO. Clearance is about the same as what we see with the 2-methyl analog. Half-life's a little bit shorter, but the Cmax is about 3-fold higher and about 2-fold higher AUC with this compound. We do see 165% bioavailability. So these things are accumulating. But we do see. I don't think there's any denial that we see very good exposure with this compound. 2043 also. I think this is a compound that shows the efflux, but interestingly has very low clearance, which again lines up pretty well with the MLM. So we're seeing this good in vitro in vivo correlation, which is encouraging and something that makes the SAR, I think, easier to follow if we can consistently show that. The half-life increases correspondingly, and the Cmax and AUC were essentially, pretty similar to what we were seeing back with 1062, 1055, and 2009. And then 2045 as well, where we've capped that an age group. This compound as well, slightly higher clearance, but again, really good exposure, certainly comparable to the original compound. And so we've moved 1062, 2009, 2043, and 2045, all 4 of these compounds into blood-brain exposure experiments. And interestingly, 1062, 2009, and 2045 all showed good brain exposure in the 3 to 6 micromolar range, and with blood-brain to plasma ratios between 0.5 and 1, roughly. The only compound that didn't show good brain exposure was 2043. This compound here, which has this efflux observation. So, I think again that's in vitro in vivo correlation. If we can demonstrate that consistently, then that will certainly help drive the SAR much faster and help us choose which compounds to move into these blood-brain experiments. So again, I'll show the absolute numbers in the experiment at the next meeting. We just, as I mentioned, we just got the data, so we don't want to show any figures at this point, but, you know, we think it looks encouraging, and, you know, I think we'll be talking about next steps with these molecules in the not too distant future. I think, you know, I also want to highlight these literature compounds that have been made here at Charles River to support the project, and I think, you know, these are some pretty exciting results, especially the blueprint compound. You know, there's no hydrogen bond donors. You know, this might be a compound that we could consider moving into an in vivo experiment to see if we have any PK. Now, let's get this in vitro data first, given that we do see a pretty good correlation. But, you know, I think that that's something that we could consider doing, because with the selectivity of that compound, the whole point of this project is to get a compound that can get into the brain and treat these patients. So if it comes from our lead series, that's great. If it comes from, you know, another series, then, you know, that'll be a discussion with Blueprint, you know, at that point. But, you know, I think let's get data and let's prove the hypothesis as fast as possible. The Novartis compound, I think, is also interesting in that it's very selective, but it does have, you know, 4 hydrogen bond donors in the molecule, and, you know, the chances of that one being brain penetrant, I think, are less likely, but, you know, I think it's still interesting, and it shows that you can be extremely selective with ALK2 inhibitors against ALK5, and getting X-ray structures of these 2 compounds, you know, may help us try to capitalize on some of that information. So I think, you know, that's something that, you know, we've talked about prioritizing. And then, you know, as David highlighted, I think there's some interesting new compounds coming out that show that we can, within this scaffold, improve the potency, you know, another 4 or 5-fold, which, you know, I think would be very, very nice, especially if we can do that while maintaining good specificity. I think, you know, these numbers are okay, but I'd like to see, you know, higher selectivity numbers there. And then, you know, I think these methoxy replacements look encouraging. We can get the intrinsic potency. Selectivity is not there yet, but, you know, I think that there's some exciting data around that as well. And then, you know, I think just some subtle variations, the magic methyl effect, you know, I mean, let's keep building on substitutions around subtle modifications around this molecule to see if we can improve it even more and try to capitalize on these SAR findings. So again, I think, you know, again, this month we've made some exciting progress. I think we continue to identify good molecules. I think some of the tool molecules that we've generated already have shown some very promising overall features that, you know, may enable some more work. And I think, you know, we need to discuss as a team what are those next steps, and, you know, what's the best way to push these compounds and to really prioritize them and to, you know, I mean, it seems like we're going to have a handful of compounds, maybe a big handful of compounds that we'll have to differentiate between. And, you know, I think we need to decide as a team how best to do that and what the most economical way, both time and money, of doing that is. And so, you know, I think that, you know, if we can get them tested in some in vivo models, that would be fantastic. I think, you know, we certainly had a meeting today, and you know, we're talking about prioritizing the PD effect, the downstream PD effect. I'm not sure why this ended, but you know, and emphasizing the effect on the pharmacodynamic markers, the phospho-SMAD, the downstream effect on the ID2, and you know, I think that's certainly some. Did we lose connection? Interesting. Angela, can you guys still hear me on that end?
Yep, we can hear you.
Can you still see the slides?
No.
Not now.
Yeah, I got a connecting to Zoom message. Anyway, I think we're at the end of the meeting. So, you know, I think we continue to generate some encouraging results and let's all continue to work towards curing this disease. So
Thanks, Jeff.
with that, I think some of the next steps here are already done now. But we're continuing to profile in DIPG lines. We want to continue to profile in brain and plasma. So we've got exposure. Let's look at what those three fractions are in each of those domains. I think more expensive kinase profiling for some of these more promising compounds as well is something that we should do, just to see what we're looking at with this 4-methyl especially. Perhaps the blueprint compound as well, if it's something that we want to see. We've talked a lot today earlier before this meeting about ways to advance the compounds and what types of in vivo efficacy studies we can move these compounds into. And I think the general consensus was that the DIPG 007 model seems reasonable based on the markers that have been shown, at least as a preliminary proof of concept. And so I think we'll move in that direction. It sounds like there may be other opportunities for additional animal models. And so, I think we'll be able to discuss this a lot more at the next meeting as we continue these discussions and then finalize some plans.
So I have a question for the drug discovery here. So we have a molecule, it gets into the brain, and we show that concentrations that it hits this pathway. We know it kills the cells. Then everyone goes, let's put it in mice. We'll create this artificial mouse with a weird ass mutation. We'll choose one cell line that's weird to put in. Why not argue that it should go into patients then? What are you actually going to learn from an efficacy experiment in mice that's going to stop you from going in? Because if it doesn't work, then you're going to start farting around until you find an experiment where it does work. Is there any rational reason?
One, I think that there is so there's a lot more that we would have to do in terms of talks, in terms of, right, before we can move something into man.
Yeah.
So to justify doing that, I think we want to consider. I agree with a lot of, especially the brain.
Alan, what happens if one compound works in vivo and one compound doesn't? Would it matter to you?
In theory, you'd feel better, but it's probably not evidence, right? You don't know that the compound that didn't work for some weird ass reason.
You're also going to have to argue with the regulator.
No, I understand, but no, regulators have the molecules into people without any animal models. That's been done in other diseases, where there is no good model and there's a clinical need. I just want to put it out there because we're like automatons, right? And it's usually because they want figure 5 in the cell paper where the reviewer goes, where's the animal model? And then you ask them, is there any proof that it's, there's a correlation? No, but I want to see it in an animal, as if some magic thing you're going to find. So I really want to find out, what are you guys going to find?
Well, hopefully, and so far what we're seeing is things correlate. I think if that were the case,
maybe not.
The original compound was actually got into the brain. It showed some effect in an in vivo model. I mean, that compound could have been moved in, right? I think we have better compounds now.
I can understand safety with the therapeutic index and the alpha 4, alpha 5. That I can understand.
Yeah.
But anyway.
No.
If you go into a safety study and you get a signal, you don't know whether there's a window over the therapeutic effect that you can see. I think it can help. It can give the regulator confidence that maybe there's a window. Otherwise, how do you pick your dose for a human study? Unless you've got a wealth of human in vitro data that you can use. So it's what dose you're going to go in at.
The dosing, but you can calculate from all your stuff that you've been doing what concentration you're getting in the brain, though, without having to do it in a model to show efficacy on the tumor there.
But you want to run it so that you get target-induced exposure and efficacy.
Why efficacy?
Because efficacy gives you your guideline for your tox. So your highest tolerable dose plus your highest efficacious dose times 3 times 10, which is your efficacy study. So that's the main driver in oncology.
Yeah, and that's a good correlation that works across all these animal models.
It'll get it through phase 1/2A because of the safety profile.
Yeah.
But then it's the efficacy in the 3D. You can't predict that.
Then you dose ranging, so you find a
Yeah.
So you
limit.
dose range in phase 1 anyway, right? Then you dose range again in people. Because the mouse.
Dose range for safety.
Yeah,
I
yeah.
recommend.
Anyway, it seems bizarre because it seems like it exists and people make excuses to rationalize why it exists.
I think a good example is the CAR T work, or how can you show what human cells going to do, but they still put it in the humanized mice. The safety is totally decomposed, but it's just, I don't know if it's just dogma and that's what we have to do, or, you know, especially CAR T raises that question, but certainly here where you've got small molecule, it's the expected.
I understand the concept, but have you ever?
Hey, this is Gene Wong. I'm one of the trialists here working with Java Nazarian, and it's a really interesting question you pose. In my mind, I think that there are there's a scientific reason to potentially do in vivo modeling, and then there's a reality reason. And from a reality standpoint, these patients, especially when you're subdividing them with particular mutations, in this case an ACVR1 mutation, are quite rare. There are hundreds of agents out there, thousands of agents that I think have some preclinical efficacy signal or some logical molecular or biological rationale behind them, and we just can't test all of those. And so in the children, so you're absolutely right that especially in the adults, there have been cases like the checkpoint inhibitors where there's been very minimal preclinical data, certainly no in vivo modeling, and then they've turned out to be home runs in the clinic. But to be able to pick out our best players is because we really have to be selective about how which ones we're exposing the kids to. And so the toxicity question, which I heard raised as a perfect reason why in vivo models are supported. Just the sheer logistics of too many drugs and too few patients. The regulatory landscape also in pediatrics is different from adults. And so definitely the CTEP and the FDA and the BMSC will all, I think, look so much more favorably if there's at least some in vivo data, not just from a toxicity standpoint, but from an efficacy standpoint. And then we as a community in the neuro-oncology side have for so long tried to figure out why our preclinical models weren't really, as you pointed out, good predictors of what is helpful in patients. And so, I think from a nihilistic standpoint, you could say that we don't need any preclinical modeling for any of these drugs because it never really helps us predict things. But the side that the community has had for many years, and maybe, I hear your argument of maybe one potential obstacle to some of the progress that could have been made is nonetheless still present, which is that blood-brain barrier penetration, microenvironmental issues, in situ, like specific locations for in vivo modeling is we at least hope more important in helping us decide what the best approach is moving forward. But I definitely appreciate your point, and I really enjoy the fact that you raised these out-of-the-box, really aggressive thoughts, which is what we have to do at some point to move the field for sure.
Thanks.
Great, thank you very much. Yeah, I think it's another question that we should bring in, have with clinicians, et cetera. Around the table, I can think of a number of reasons, but not being a clinician, I'm not going to stick my neck out on this, but having gone through it, I can see it might be very hard to attract clinicians to a study if they think, it's very different model. We brought in a very different mechanism of action. We had a lot of skepticism with docs about enrolling their patients into our study. And, you had to prove that your mechanism of action worked, et cetera, to get the docs to do this. If the doc's looking at enrolling his patient into different studies, they may take the view of whichever one is the most scientifically supported by the various things, which is another reason for why you go forward with as much data as you can to encourage people to go into that study, which everyone believes is the highest probability of success, regardless of what we may or may not think of the actual mouse animal models. They are the best thing you have. And that's enough from the finance guy. I think we should wrap it up now, unless there's any other questions. But I think this is a very interesting slide. This last one that's been up here reminds you that no matter how much you've actually done, there's actually twice as much to do every time you come up with something. So this is welcome to the story of drug development. Sorry, was there one last point to bring up there?
No, but we're in proof of concept development, right?
Understood.
I mean, at some point, I'm not smart enough to do it, but you guys could decide when enough's enough, right? Because perfection is the enemy of the good, and we really want to test the hypothesis that inhibiting this kinase is good in kids. And so continue to optimize and get something else. I didn't even get pickleball. Yeah.
And again, I think it's a good point to bring in clinicians to get their feedback, because, you know, it's one of those things where you can start running off, as you say, doing the scientific, you know, keep playing with something too long, and say when the clinicians are saying, well, this is much better than anything else I have available to me. So maybe, you know, start talking about that. But anyway, that's for another question. I think it's a good reminder that we need to make sure that we have clinicians involved in these conversations at an earlier stage.
Yeah, I think, you know, because it's a kinase, I mean, selectivity, specificity is going to play into